dive in a little more into a particular blood type. How common is type O blood?
So type O blood in total represents about 45% of people in the US population, but O positive.
For centuries, type O blood was seen as just another medical fact, a universal donor, useful in emergencies but otherwise unremarkable.
Patients who have type O, they have no antigens on their surface, which is why type O can be the universal donor.
Yet, the true story hidden in our veins is far stranger. Millions of people across vast continents carry a blood type woven with the legacy of survival and mysterious ancient trials. How did nearly entire populations in the Americas come to share this genetic signature while others vanished? What ancient forces erased competing blood types? And what secrets does type O carry deep within its code? The puzzle of type O dominance. Type O blood is a familiar term in medical clinics worldwide, often labeled simply as the universal donor, a lifesaver in emergencies when the perfect match isn't available. But beyond this practical use lies a genetic mystery far deeper and more complex. In the Americas, particularly among indigenous populations, type O blood isn't just common, it is overwhelmingly dominant. In some Amazonian tribes, close to 100% of individuals carry type O, a uniformity so extreme it defies casual explanation. This prevalence stretches from the icy tundra of Alaska down to the dense rainforests of the Amazon. Arctic hamlets and isolated Andean villages also display this near-complete dominance, passing the same genetic trait through generations with remarkable consistency. This pattern isn't scattered or occasional, it's a continent-wide echo resounding through millennia. Such overwhelming uniformity raises a crucial question. Why does type O blood stand so singularly normalized across these diverse and often isolated populations? Scientists initially attributed this dominance to well-known mechanisms in genetics. Isolation and founder effects, where small populations pass on limited genetic variation, seemed a reasonable explanation. In tiny groups cut off from outside contact, random genetic drift can magnify specific alleles, allowing certain traits to dominate purely by chance. If the first settlers of these regions carried mostly type O blood, the resulting genetic legacy could look like what's observed today. Yet, this tidy explanation starts to falter under closer scrutiny. The consistency and purity of type O blood across such vast and ecologically different regions do not align well with simple drift. One would expect some pockets of variation, traces of types A or B, to survive in at least some groups. Instead, many populations show near total absence of these blood types, suggesting that chance alone cannot account for the pattern. Even in populations with documented contact or interbreeding with outsiders carrying diverse blood types, type O remains stubbornly dominant. This suggests that either there was a powerful selective advantage to carrying type O, or some other force was at play actively removing other blood types from the genetic landscape. The more data collected, the harder it becomes to fit these patterns into conventional genetic models. This bewildering phenomenon hints at a past filled with genetic disruption, survival challenges, and perhaps biological pressures that shaped who could endure and who couldn't. Type O blood, once viewed simply as a clinical label, suddenly appears as a marker of ancient resilience written in DNA. But if this is true, what exactly happened to the other blood groups? And when did this decisive genetic shift occur? These questions are far from academic. They strike at the core of human history and migration narratives. The dominance of type O blood asks us to rethink how populations moved, adapted, and survived in the New World long before recorded history. It's a story sometimes overshadowed by complexity, but too significant to ignore. As the puzzle sharpened, researchers turned their gaze to the established stories of migration, hoping that the answers to this blood-type riddle might reshape our understanding of humanity's journey into the Americas. The migration theories surrounding the first peoples of the Americas suddenly carried new weight under this lens. If type O dominance was not merely coincidence, then ancient bottlenecks, disease waves, or selective survival events may have sculpted entire populations with brutal efficiency. Hidden within blood itself could lie silent evidence of catastrophes, adaptations, and forgotten chapters of human endurance. If the origin story of indigenous blood types was more complex than simple drift, the clues would lie deep in the ancient genetic record awaiting discovery. Genetic drift or something else? The concept of genetic drift offers a comfortable framework for explaining limited genetic diversity. Small, isolated populations often experience random shifts in allele frequencies, a process natural and well documented in population genetics. It seems plausible that early groups settling in remote parts of the Americas saw type O blood dominate simply because the initial migrants carried primarily those alleles. Over centuries, isolation could reinforce this uniformity. However, as more genetic data began arriving, the drift hypothesis started to look incomplete. The level of type O purity observed across numerous distant and ecologically different groups, where various blood types should have crept back in through even limited contact, defies the randomness expected from drift alone. The lack of type A and B in many areas is too stark to be coincidence. In fact, some populations show nearly complete absence of any blood type other than O, even when living near or interacting with groups known to carry diverse blood profiles. This suggests a selective pressure or barrier, something that actively favored or preserved type O alleles while reducing or eliminating others. The data, therefore, hint at an intentional genetic filter rather than mere random genetic fluctuation. If drift were the only driver, scientists would expect at least some pockets of alternative blood types to hang around. Those leftovers would linger because complete allele extinction across entire continents rarely happens with drift by itself, like not often. This mismatch pushed the scientific community to look at the genetic pressures underneath type O's dominance. The thought that type O blood's persistence might be about a survival edge started to get real attention. Could environmental cues, immunological patterns, or pathogen-related pressures have sculpted these populations by essentially rewarding type O carriers? If yes, then this would count as one of the clearest examples of natural selection acting on human blood groups in recent memory, or even across deeper history. Still, some people argued the dominance could be explained through older population bottlenecks, where catastrophic events cut down numbers and left only certain slices of variation alive. These bottlenecks can enhance specific alleles, but when exactly they happened and how big they were is still kind of unclear. Would such an episode have leaned things toward type O, or would it simply shrink diversity in general? Yes, genetic drift and bottlenecks definitely matter, but the sheer magnitude and the geographic consistency across the Americas seem to point to something sharper at work. The idea of a survival trial, a strong environmental or biological threat acting like a sieve on human genes, began to surface. Researchers figured that to actually settle the question, they would need to look past modern populations and into ancient DNA, comparing what came before with what remains today. That approach would either support drift's more gentle influence, or it would uncover a much more dramatic narrative. What ancient DNA eventually revealed would challenge assumptions many researchers once considered stable. Buried within old remains were clues suggesting that blood group diversity in the Americas may once have been broader than today. If true, then something happened after those migrations, something powerful enough to reshape the genetic signature of entire civilizations across generations. When ancient remains finally yielded their genetic secrets, the established story began to crack. The pure drift theory simply could not hold against the new evidence waiting in millennia-old bones. Ancient DNA challenges, migration theories. For decades, the dominant theory of human migration into the Americas involved a relatively simple story. During the last ice age, small groups crossed a land bridge known as Beringia between Siberia and Alaska. These pioneers carried with them a subset of Siberian genetic diversity, including blood types, and over millennia, their descendants spread across 2 continents. It was assumed that the founding populations had limited variation dominated by type O alleles. Recent advances in ancient DNA extraction and sequencing have allowed scientists to test this assumption directly. When ancient Siberian skeletal remains were analyzed, the results surprised many. Instead of a narrow genetic pool dominated by type O, ancient Siberians showed a rich mix of blood types, including A, B, and O in significant proportions. much like populations in Europe and Asia. This diversity shattered the idea that only type O carriers made the journey into the Americas. The genetic starting point was broad, not limited. If these were the ancestors of Native American populations, their blood types should have been similarly varied, at least initially. Yet, the modern dominance of type O blood in the Americas showed a glaring disconnect. The question became urgent. Why did blood types A and B virtually disappear after crossing the land bridge, while type O surged to near exclusivity? The disappearance was not a gradual fade expected from random drift. Genetic records suggested a bottleneck event selectively eliminating A and B alleles from much of the population, leaving type O as the overwhelmingly dominant survivor. This bottleneck appeared sudden and widespread, affecting vast geographic areas synchronously. It couldn't be easily explained by climate or landscape alone. Instead, it pointed toward strong selective pressure, possibly a pathogen or environmental factor that drastically influenced survival rates based on blood type. Such a selective bottleneck implies that the journey into the Americas was more than migration. It was a crucible of survival, where genetics determined who lived and who did not. The story of human arrival in the New World now had a shadowed chapter of catastrophe and resilience. These revelations forced a rethink of how population genetics and human movement intertwine. The presence of ancient blood type diversity in Siberia, coupled with near-total loss of that diversity post-migration, suggested that the Americas experienced unique biological pressures shaping its peoples. As scientists dug deeper, they realized this genetic mystery was tied to immune function and survival, leading to the next step in uncovering what gave type O blood its extraordinary advantage. What emerged next pushed the mystery beyond migration patterns and into the biology of disease itself. Researchers began noticing that blood types influence how pathogens attach, spread, and trigger immune responses. If ancient epidemics swept through early American populations, individuals carrying type O blood may have possessed subtle advantages invisible to history, yet decisive for survival. The secrets locked in ancient genomes held the key to understanding both human survival and the mysteries behind the blood types we see today. The disappearance of A and B alleles. The story of type O blood's dominance deepened when scientists examined ancient remains across the Americas and noticed a startling pattern. Types A and B blood virtually vanished. Early people who carried diverse blood types, including A and B, seemingly disappeared, leaving only type O in vast regions.
O blood type actually looks like it's protective. It's not a huge increase in protectivity, but it's 9 to 18% less likely to be infected and be severe.
This disappearance was not gradual or isolated. It was abrupt and widespread, raising profound questions. How could such major blood types be wiped from the genetic landscape almost completely? This pattern was observed consistently from North to South America. Ancient skeletal remains from Alaska included traces of type A alongside type O, marking the presence of blood type diversity among the earliest inhabitants. However, as researchers looked further south, signs of blood types A and B dwindled sharply. By the time remains from early Andean populations were analyzed, these blood types were entirely absent, replaced solely by type O. Such a pattern challenges the traditional model of slow and random genetic drift. Typically, genetic variation diminishes gradually over time, especially within isolated groups, but rarely disappears entirely on a continental scale. The across-the-board loss of A and B alleles strongly suggests the influence of a powerful selective event or force, not chance. Something remarkable, and likely catastrophic, must have occurred to cause this genetic purge. One of the most plausible explanations involves a sharp bottleneck, a rapid reduction in genetic diversity caused by a significant population decline or selective pressure. This bottleneck would have disproportionately affected individuals with types A and B blood, rendering them less likely to survive and pass on their genes. Only type O carriers, by some advantage or circumstance, maintained a genetic foothold. The causes behind this bottleneck still feel speculative, but terrifying in how big they could be. Some scientists say an epidemic, or a string of diseases, might have been right at the center of it all. Certain pathogens recognize and then latch onto particular blood group antigens, so if a disease was targeting A and B markers, it could have wiped out whole populations. In that case, people with type O blood might have had a relative immunity, meaning they could survive, then eventually repopulate, with less trouble than everyone else. Then there are the other ideas too, the ones that point toward some unknown virus, bacteria, or even environmental disaster, like maybe more violent climate swings or nutritional shortages that somehow hit certain blood types harder. The common thread is that each version, or maybe each variation, seems to boil down to a selective force that was strong and picky enough to reshape the genetic makeup of entire groups in a comparatively short span of time. This genetic bottleneck isn't just a biological afterthought. It's portrayed more like a pivotal event that basically redirected the demographic and immune story of the Americas. And it reminds us that survival back then was, in practice, precarious. Often it was decided by factors you couldn't see, yet they were lethal anyway. The pressure was so immense that only a few traits managed to hang on. When we look at it this way, we may have to reconsider what human genetics actually means, and especially blood types. They aren't only inherited traits you pass down, they are like survival records, written by hardship. The disappearance of the A and B blood types doesn't look like random drift alone. It reads more like a genetic signature of endurance and adaptation in a rough prehistoric world that didn't forgive much. The deeper scientists explored these vanished alleles, the more they realized the mystery extended beyond genetics into immunity itself. Blood group antigens are not passive labels. They influence how viruses and bacteria recognize human cells. If ancient diseases selectively targeted certain blood markers, then entire communities may have been reshaped by invisible biological warfare unfolding over generations. From here, the question shifts to what those survival advantages were, the hidden forces that made type O a genetic lucky key through this ancient trial. The survival trial theory. The notion of a survival trial draws together the fragments of mystery surrounding type O blood dominance. This theory suggests that during early human migration and settlement in the Americas, a powerful selective filter-shaped blood group prevalence favoring type O carriers. It wasn't just survival by chance, it was survival by blood. Pathogens are among the most likely culprits behind this selective pressure. Some infectious agents exploit blood group antigens to enter cells, and if particular diseases specifically targeted A and B antigens, type O individuals might have been relatively protected. This would grant a decisive biological advantage during epidemics, allowing them to survive waves of infection that devastated their neighbors. The idea of a survival trial also aligns with evidence from other parts of the world, where blood type plays a subtle role in disease susceptibility. Though no blood type guarantees immunity, variations in infection rates among blood groups, from malaria to cholera, indicate that blood serves as a frontline battlefield in the ongoing conflict between humans and pathogens. In this theory, the dramatic reduction of type A and B in the genetic record wasn't merely a side effect of drift, but a consequence of a harsh, targeted event, a catastrophic episode or series of episodes that winnowed the population genetically. The survivors carried type O blood, bearing its genetic advantages like a biological badge of endurance. This survival filter would have profound impacts on the descendants of those early carriers, propagating a genetic legacy that echoes in modern populations. The near-total prevalence of type O blood in indigenous American groups isn't just coincidence, it's a result of being the genes of the most robust in a severe prehistoric trial. Importantly, this process would have been rapid and decisive, not a slow march of mutation or chance. The near-simultaneous loss of diverse blood groups suggests that the populations faced a shared challenge with devastating consequences, cutting deeply into the gene pool in a historically narrow window. Other selective forces, such as environmental stressors and nutritional challenges, could have coalesced with infectious pressures, compounding the survival odds. Taken together, these pressures forged a genetic bottleneck, compressing diversity while imprinting a survival advantage onto those with type O blood.
Most of the world didn't have it until colonization started in the 15th century.
And it's the one blood type that is least likely to mutate or interact with other blood types.
Scientists now see this survival trial as a critical chapter in human evolution within the Americas, a natural experiment with lasting genetic signatures. It reminds us that human survival is fragile and shaped by the ever-shifting threats posed by our environment and pathogens. This understanding also humbles present-day us. The genes we carry are not just legacies, but survival stories written in the fabric of our biology. Stories that continue to influence health and disease centuries later. From survival trials, the inquiry naturally progresses to investigating what biological mechanisms underpin Type O's advantage, guiding researchers into the complex interplay of genetics and immunity. Immune system links and genetic variants. As researchers looked closer at the survival edge Type O blood seemed to carry, they began to uncover deeper connections to the immune system, links that went beyond blood group antigens to underlying genetic factors influencing disease resistance. One gene variant, known as IL-17 variant 52C, emerged as a particularly striking piece of this puzzle. Found at unusually high frequencies in indigenous populations living in some of the most challenging environments on Earth, this variant appears to fine-tune immune responses in subtle but critical ways. The Andes Mountains provide a vivid example. Despite the thin air, intense ultraviolet radiation, and a long history of infectious disease exposure, many Andean communities exhibit remarkable long-term survival and health. Genetic studies revealed a high prevalence of IL-17 variant 52C in these groups, notably linked to Type O blood. This variant seems to act like a biological thermostat on the immune system, regulating inflammation to prevent the deadly overreaction that often proves more harmful than infection itself. In many severe illnesses, death results not from the pathogen alone, but from the immune system's excessive response, a phenomenon called a cytokine storm. IL-17 variant 52C appears to moderate this risk, helping the body balance fighting infection with protecting its own tissues. This genetic combination could represent a finely tuned evolutionary adaptation forged in response to centuries of disease pressure in harsh environments. The correlation between Type O blood and this immune variant is far too strong to be coincidental. It suggests that populations which survived environmental and pathogenic challenges over millennia did so in part because they carried this genetic advantage. Those who lacked the combination may have been more vulnerable and less likely to pass their genes forward. This ongoing selection pressure might explain why Type O blood and the IL-17 variant have gotten so tangled up in indigenous gene pools. It's a clear example of natural selection happening, like survival of the individuals that were best genetically equipped to push through recurring threats. Over countless generations, this back and forth has left a genetic imprint that mirrors those ancient struggles. Still, these findings mostly stay in specialist genetic journals, and they're sometimes missed by mainstream media or public health talk. The notion that a blood type could carry sort of hidden traces of older pandemics or survival trials quietly reframes how we think about human biology. It nudges awareness that genetic resilience is often a narrative written way before modern medicine even shows up. Even so, this isn't a cure-all, or some sure thing immunity shield. The mild advantage from these genetic factors might have mattered a lot when medical interventions weren't around. Today, Type O carriers may still gain benefits in ways science is only starting to map, a living reminder of the evolutionary battles their ancestors had to face. The links between immunity, genetics, and blood type show biology as this layered, complicated survival system. They remind us that our genetic makeup is an evolving record, not merely of who we are now, but of the challenges humanity kept running into across time. As researchers continued to unravel this genetic tapestry, they discovered an even older clue, a mutation frozen in the permafrost that opens a window to the distant past and points to an unbroken lineage still alive today. The ancient O-delta variant. Deep in the frozen soils near Siberia's Lake Baikal, archaeologists uncovered skeletal remains dating back more than 12,000 years. These remains opened a rare gateway into the genetic past of humanity, revealing a surprising detail: a rare deletion mutation in the blood group gene, now called O-delta variant 1. Unlike typical genetic mutations involving single-base changes, this deletion removed an entire sequence of DNA, a sign of intense evolutionary pressure on this lineage. What makes O Delta variant 1 extraordinary is its exclusivity. It is absent in most ancient and modern populations across Europe, Africa, and East Asia, appearing almost solely among certain indigenous peoples of North America. This suggests it marks a unique genetic fingerprint linking ancient Siberian ancestors directly to the first peoples of the Americas. It's a thread tying together generations across tens of thousands of years. The functional implications of this deletion point to changes in how the immune system might recognize and respond to threats. Its presence implies that the populations carrying this variant endured an intense biological struggle that forced their genes to adapt in drastic ways. This was not a benign mutation, but a survivor's scar etched into the human genome. For anthropologists and geneticists, this discovery frames type O blood not merely as a common blood group, but as one carrying a legacy of hardship and resilience. Those who bore the O Delta variant survived catastrophic events that erased other lineages, passing on a durable genetic code adapted to survive extreme challenges. This survival was no accident. O Delta variant 1, tightly linked to type O blood, indicates a lineage preserved through millennia of climate upheavals, migrations, and emerging epidemics. It embodies an evolutionary triumph amid relentless pressure. The puzzle shifts our understanding of migration and survival, showing that the story isn't simply about ancient peoples crossing land bridges. It's about who survived what came after, shaping the genetic landscape we see today. It reframes ancient migrations as Chapter 1 of an ongoing battle for survival fought at the microscopic level. The variant also serves as a reminder that human adaptation goes beyond visible traits, weaving into the molecular fabric of our cells markers of resilience that science is only beginning to trace. This remarkable genetic thread connects Ice Age Siberians to present-day indigenous peoples, a living bridge spanning millennia and continents. It hints at a deep, enduring immunological heritage carried quietly through generations. From here, researchers moved toward the present day, uncovering startling new evidence hidden in modern DNA samples. The Crawford revelation and global links. In early 2025, an unexpected discovery emerged from a genetic database, sparking excitement and curiosity among scientists studying type O blood. Thomas Crawford, a resident of the Blackfeet Nation in Montana, volunteered for an ancestral mapping project and unknowingly became central to a breakthrough. His DNA carried the rare O Delta variant 1 deletion, connecting him to ancient Siberian ancestors who survived intense biological pressures thousands of years ago. This alone was remarkable, but something even stranger was uncovered alongside it. Near the human leukocyte antigen region, a critical zone for immune response regulation, researchers found an unusual genetic fragment in Crawford's genome. Previously dismissed as junk DNA, this segment revealed itself under advanced immune simulations as a potential emergency defense switch. Silent under normal conditions, it activates during extreme biological stress, such as widespread infection or oxygen deprivation, regulating immune cascades and protecting vital organs from collapse. This hidden genetic mechanism is unlike anything scientists had seen before. It appears to function like an ancient backup system embedded deep within certain human genomes. Its presence was disproportionately associated with type O blood carriers from some indigenous populations, often alongside the O Delta variant 1 mutation, suggesting a linked survival strategy evolved over millennia. The discovery was thrilling, but also shrouded in mystery. Evidence surfaced that this fragile clue was initially identified decades ago in 1998, during early human genome sequencing efforts. At that time, its purpose was unknown, and strangely, all public records and research references disappeared. Some insiders hinted the knowledge was deliberately suppressed, possibly due to its controversial implications about engineered or unique survival traits in human bloodlines. Whatever the reason for this silencing, the fragment re-emerged at a time when ancient DNA research and modern genomic tools were finally mature enough to explore its meaning. Its connection to type O blood's extraordinary resilience tied together ancient survival events, genetic legacy, and new questions about human adaptability. Meanwhile, genetic analyses extended far beyond North America. Researchers tracing type O's genetic trail discovered links spanning the vast Pacific Ocean to Polynesian populations. Skeletal remains from Rapa Nui, or Easter Island, dating back roughly 900 years, revealed not only overwhelming prevalence of type O blood, but also the same IL-17 variant 52C immune gene found in Andean populations thousands of miles away. Such a close genetic match across vast oceanic distances indicates direct biological exchange, suggesting pre-Columbian contact between Polynesian voyagers and South American peoples. This implies more than cultural or trade connections. It points to the transfer of vital genetic tools adaptive for survival in the face of deadly diseases and harsh environments. Further reinforcing this narrative, archaeological studies in the Andes revealed ancient dental enamel containing antibodies tuned to pathogens long extinct today. Nearly all these immune traces belonged to individuals with type O blood, while other blood types were almost entirely absent, likely because their carriers did not survive repeated epidemics. Type O therefore emerges not just as a blood group, but as a living record of enduring hostile environments and devastating disease outbreaks. These revelations sketch a sweeping portrait of type O blood as a genetic signature shaped by survival trials, migrations, and even long-distance oceanic voyages. It is a marker of both collapse and resilience, a lineage that has endured when countless others vanished.
O positive, O negative, that can freely give to all the blood types. So we're talking about blood type O. And how I remember that blood type O universal donor.
Type O blood may be humanity's ultimate survival code, an unbroken thread woven through time, climates, and continents. Type O blood is far more than a universal donor. It's a genetic echo of ancient survival and resilience. From Siberia to the Americas and across the Pacific, its story challenges everything we thought we knew about human history. If you found this journey into blood's hidden past fascinating, be sure to like and subscribe for more deep dives into the science and stories shaping our world.